Okay, so thank you very much for the kind introduction and thanks for the invitation. I'm very happy to be here and see so many familiar faces. I have many collaborators or colleagues in Zurich, actually. So, well, we will talk a little bit more about the blue guy you have on your name tag here. So bacteriophages, indeed. And I will focus, as she just said, about the selection and production of antimicrobial agents that can spare the microbiome and especially phages for phage therapy. So very quickly, phages, you all know that they are bacterial predator in nature and they are the most abundant entity on our planet. Okay? So what is very interesting already here when you think about treating patients with phages is that there is actually no human disease which is associated with the phage particle itself. So this is something telling you that it's quite safe, we think. So then it's very interesting, those phages, because some phages are able to kill some multi-drug resistant or pan-drug resistant strains, so it's quite interesting nowadays with this crisis of antibiotic resistance. And what is very interesting but brings a very high complexity when you want to produce them and select them is that they are very specific for the species they infect. A Pseudomonas phage will not kill a Staph aureus strain. And they are also very strain-specific, meaning that when you have a phage against Pseudomonas, it will kill one strain, but not the other one, maybe. The coverage in Pseudomonas is like 40% of the strains for one phage. Acinetobacter baumannii, we have example where we have one phage that can kill one strain on 100. Okay, so you can start to imagine how it's difficult to tailor the treatment for the patients. Here you see the nice picture of a phage and the bacterium. How is it working, this? Yeah, like this. So this is a phage and this is the bacterium. So now, phage therapy, which uses phages to treat patients, is a very highly specific treatment. It's a very elegant treatment of bacterial infection, which again spares the commensal bacterial flora, because here you have the example of antibiotics, which will actually target the pathogen, but also the commensal flora, and eliminate both, usually. Here, bacteriophages, they will perform a targeted attack on the pathogen, replicate on it, and then kill it without harming directly the commensal flora. So this is the main advantage of phages over antibiotics. Now, how do we, first of all, produce phages? We have two steps. I will explain you the first step, which consists in producing concentrate of bacteriophages that we call CBPs. And why do we go through this first step? It's because these CBPs are very highly concentrated and they are very stable. But you have a lot of contaminants in those CBPs, like endotoxin, for instance, which I think helps for the stability. Still, these lysates or CBPs are produced in GMP, so good manufacturing principles, and these are the standards, the principles that you need to apply when you want to produce a medicine. And phages are classified by Swissmedic as biological medicines. So you need to produce phages as any other medicine if you want to give them to patients, at least in frame of clinical trials. This is what we plan to do at the CHUV. Okay? So here you see a picture of my lab where we have a dedicated room and material, equipment, everything to produce these CBPs. And you see how it goes. We have production strain. We still think that we could produce the phages on the patient strain, which would be actually very useful because of the specificity, but sometimes the strain of the patient does not grow well, so it's tricky to produce the phages on them, so it's not a rule, a universal rule. And then you mix this production strain with your phage from the bank that you have assembled into a large volume. We use 1 liter and in growth media. And then after several centrifugation, frontal filtration, again centrifugation, we have in total 3 centrifugation, as you see. We can get this concentrate of bacteriophages, which is at 10 to the 10 to 10 to the 14 phages per ml. So very high concentration. And this, we store them in saline at 4 degrees, and you see we have stability data right now for up to 1 year. The titer does not move, actually, which is very good news. So then, as soon as a patient is coming at the CHUV, we get the strain from the microbiology at the CHUV. And here we are doing a phage selection step, what is called a phagogram, like an antibiogram. And we do that using a DDT, for diluted drop test technique first, in order to quickly see which phages are active. So we have collections for Pseudomonas of more than 100 different phages, so which phage should we use for the patient, okay? So we will test our CBPs, which are pre-prepared, on the production strain that we are using, and also on the patient strain using these tests where you put the strain into a soft agar which solidify at room temperature, and then you deposit the phages, 5 microliter on top of the loan, and you dilute 10 by 10, and then you get this kind of patterns. Here you can see small dots, you can count the phage and get the titer, so we can calculate the NEOP score for efficiency of plaquing, which is the titer you obtain on the patient strain divided by the titer on the reference strain. You can even score the lysis, which is clear, semi-clear, turbid, and so on, or no lysis. So here then we are reporting this score in a table like this, and we get the score, okay? And then, it's not the only test that we are doing, then we check if the phage are able to inhibit the growth of the cells in liquid. In a simple kinetic assay, turbidity assay, we are, so here in black you have the strain of the patient which is growing alone without phages, and then we add the phage, and here we have 4 different CBPs, C4000, 4001, 2, and 3, and you see that these 2 are working very well because they inhibit the growth of the cells during 24 hours, while the other one are not working. Okay, so here we say, well, we should go for 4001 or 4003. And then comes the second step. On demand, let's say we will produce C4001 for this patient, then we dilute this stored CBP to a concentration of 10 to the 8 and 10 to the 9. And this is because when you have high titer, you have high endotoxin, and it's tough to remove the endotoxin. Because you need to reach a certain threshold, 5 units per kilogram per hour, if you want to inject IV medicine to a patient. Okay? So you need to decrease quite extensively the endotoxin, which is 1 million units per ml in those CBPs. Okay? In order to do this, so we dilute, and then we deliver this to another facility at CHUV, which is called the CPC, for Center for Cell Production. There, they have already a GMP accreditation from Swissmedic to produce advanced therapy medicinal products, ATMPs. So they will prepare the final product, which is called the SBP for suspension of bacteriophages, according to the GMP. It's tough, huh? CBP, CPC, SBP. That's life, okay? We had to think about it a lot. Still, you see the facility here. They are using isolator, where they are actually producing the SBP according to the pipeline we developed. Again, filtration, they use some specific resin to remove the endotoxin, and then they aliquot this in flask of 1 ml, which is 1 dose, okay? And they release this to the clinic. I will go quick on this one. We have a lot of quality controls that we needed to develop and validate with many partners, and you see that most of them are done on the SBP. We sequence the full genome to be sure that we produce the right phage, which is not bad to check, the activity on the patient strain, and so on, phage titer, endotoxin level, and so on. I am very proud to tell you that we were able to release the first batch, validation batch for the marketing authorization that we are submitting to Swissmedic back 1.5 years ago, and we did a lot of work to adjust things also in between. And now we are ready in the coming weeks to submit the marketing authorization for the clinical study to Swissmedic. So here I show you just the outlook of one module on 5 about the quality of the product. So here you see, you do not see CBP, because CBP is also called phage CHUV to make things simple. Okay, so you have to explain all the process, everything, how we purify, why we use these quality controls, and so on. Same for the phago CHUV, which is the final product, and so on. So I will stop here because Robin told me that I have no time anymore. And I thank the Fagochuv team, the main actor, many people involved in different places, the CPC, the EMUL, the pharmacy, and the proteomic facility of the UNIL also. My technicians, who are doing an amazing job to prepare, set up all these things. Setting up GMP principle in a research lab is not something very easy, I can tell you. And the funding, of course, and you for your attention. Thank you very much. Yeah.
Do you hear me? Yes.
Yes.
Thanks a lot for the presentation, Greg. With the amount of phage that you are administering, do you ever see an immune response?
Yeah, this actually, we know that from animal studies in my lab, because we did that in animals for the moment. We didn't treat patients yet. But it's known that we produce antibodies against phages. Many groups are working on why is it still working. On all, and this is Georgia Mitropoulou at CHUV, made an exhaustive review of the human cases for the Swissmedic file. And it turns out that in only one case, they found a correlation with antibodies presence against the phage and failure of the treatment. In all others, there is no link. Maybe affinity for the substrate, and things like that, competition. Yeah.
Do you plan to do phage training to increase the efficiency of your phages?
Good question. I talked to Martin Lössner, all Martin Lössner in Zurich, a lot about this, because we can modify phages genetically to improve phages. But then you enter in the GMO field, and which is very difficult actually to tackle. I think phage training is the way to go. And this is what Daryl said in the 20s. So I really agree with Daryl. Yeah.
We have a question over here.
Yeah.
Fascinating to see application of phages. How do you deal with the emergence of resistance for the bacteria?
Good question. For the moment, because of the complexity, we are treating patients, we plan to treat patients with a single phage, with a curve that is mostly flat. But what we do, and what I didn't show, is that we are systematically starting to study resistance that we select in vitro. So we test the virulence in Galeria melonella models and so on, just to see, and there is no universal rules. The main resistance in Pseudomonas loses the LPS, which is the receptor for the phage, and then you do not find it in vivo because it cannot infect anymore. So this is a very great question, but we have a lot of work on that to do.
We have a last question there.
Does this work? Here we go. Okay, my question is related to a statement that you made in the beginning of your talk about how there's no indication that phages are important for can cause damage to human cells. And I think that's largely known. So they're very distinct for being targeting to bacteria cells. But we know that bacteria really support human health, and a lot of this research has only come up in the last 20 years. And a lot of it has been largely ignoring phage biology. So my question to you is whether we know if there are some phages that are very harmful to beneficial bacteria and in that way are harmful to humans.
This is why I said no direct effect. This is on purpose. But still, I didn't read up to now dramatic stories in patients. So I think it's quite safe, but of course you will have release of the entire content of the pathogen in a microbiome. So maybe, and for sure, you have side effects, collateral damage. But I think it's less than antibiotics still. And also we will use phages with antibiotics, in combination, because they are usually very synergistic. So we will not use phages alone for the moment. But this is something going on in the field also. Yeah.
Thank you very much.
Thank you.